Hello everyone. The topic for my presentation is mechanistic understanding of in vitro drug release from long-acting injectable suspensions. Long-acting injectables are the drug products those are designed for the controlled delivery of active pharmaceutical ingredients. These suspensions contain highly hydrophobic API that is usually stabilized by polymeric or surfactant stabilizers. They have number of advantages such as improved patient safety, less pill burden, improved patient adherence, and reduced cost of treatment as patients do not need to make frequent visits to the clinic. These drug suspensions are believed to follow Noyes-Whitney law for the drug dissolution. However, this may not be true as the particles are retained as depot at the injection site. So whenever these drug products are given at sub-Q or IM site, they form a depot from where the drug is released slowly. And it is believed that the inflammation at the injection site play a very important role in governing the drug release, initially through the increase in tissue volume and later by the formation of granuloma around the drug particles. So during the granular formation, there is a likelihood of these particles undergoing agglomeration and affecting the drug release. The current drug release method, which is the USP type 2, does not take this into consideration as the particles are dispersed throughout the release media. Hence, there is a need to develop a release method in which a suspension can be retained in the form of depot while performing the release studies. So the objective of this research was to develop a novel dissolution method for long-acting injectable suspensions to investigate the importance of particle agglomeration in the formulation development of LAI suspension, and lastly, to study the impact of different particle states on the in vitro drug release. We started with preparing different Q1, Q2 equivalents for the Depo-Provera injection, which is a long-acting contraceptive. So we prepared 5 formulations, 2 were prepared with formulation changes, and 3 were prepared with manufacturing changes. So for formulation changes, all the excipients and the particle size of MPA API was kept same, except the PEG source, which was obtained from Spectrum Chemicals for formulation FA and BASF Chemicals for formulation FC. For the other 3 formulations, we changed the particle size. In case of FP, we use anti-solvent method and try to increase the particle size. And in case of FD and FE, we reduce the particle size by high shear techniques such as homogenization and wet media milling. So for the in vitro release studies, we performed the dissolution using a novel adapter that can be used in USB type 4 apparatus for performing the release study. The advantage of this adapter is it can help to maintain suspension in the form of depot while carrying out the release studies. So on the picture on the right, we can see that how suspension is retained as a depot into the conical cavity of the adapter. We did some low energy sonication experiments to determine the effective particle size and to see whether there are particle agglomerates in the suspension. So when we applied low energy sonication while doing particle sizing for formulation RLD, FA, and FC, we can see there is a decrease in particle size over time, suggesting that particle agglomerates are breaking with the application of sonication energy. And these suspensions were termed as suspension with weak agglomerates. For the other formulations, we can see that on application of sonication energy, formulation FD shows slight reduction in particle size, whereas FD and FE does not show any decrease in particle size, suggesting that these suspension may be with these strong agglomerates or with the primary particles. So to confirm this, we performed some microscopy experiment. And from the microscopy data, we can clearly see that in case of FB, there are strong particle agglomerates. And in case of FD, there are particles present in the form of native state, also called as the primary particles. So from this study, we concluded that in case of suspensions, the particles could be present in the form of 3 different states as primary particles or particle polymer agglomerates or particle-particle agglomerates. And now we went on to investigate like what would be the impact of each of these states on the in vitro drug release. So we started with particle polymer agglomerates. So as I discussed before, these formulation FA and FC are the suspension with similar particle size with the only difference in the PEG source. Even after having similar particle size, they showed marginal differences in the drug release. And to understand these differences, like what could be the attributing factor for these differences in the drug release, we carried out experiment wherein the particle size was determined using pipette and syringe. So we introduced our suspension sample into the particle sizing system using pipette and syringe and check for the particle size for formulation FA and FC. So in case of FA, we can see that when the suspension is introduced using pipette and syringe, there is not much difference in the particle size. However, in case of formulation FC, we can see there is a drastic drop in the particle size when the sample is introduced using syringe, suggesting that The particle agglomerates in the formulation FC were much more sensitive to shear, and that could be the attributing factor for the marginal increase in the drug release. So from this study, we concluded that the suspensions prepared with polymer obtained from different vendor sources could result in the formation of weak agglomerates of different strength, and that could affect the drug release. Next, we wanted to determine what would be the impact of particle-particle agglomerates. So when we carried out the release studies, as I discussed before, that formulation FB is of larger particle size. And from the release data, we can see that even after having such a large particle size, the decrease in release rate was not much. And there was not much discrimination between the different formulations. So we believe that the drug release from this formulation is faster than the expected drug release. So to understand this, we determined the impact of release media, wherein since the release is carried out in sodium dodecyl sulfate, what would be the impact on the particle-particle agglomerates of this media? So from the particle size data, we can see that at day 2 and day 4 of dissolution, there is drastic drop in the particle size, suggesting that sodium dodecyl sulfate might be having a stabilizing impact on these particle-particle agglomerates, allowing these particles to rapidly deglomerate and increase the drug release rate. So to prove this, we change the SDS media by PBS, and in the PBS, we can see that there is not decrease in particle size, suggesting that SDS was having a significant role in stabilizing these particle agglomerates. To further confirm this, we did a microscopy experiment wherein we can see that initially the particles are present as particle-particle agglomerates, and on day 2 and day 4 of dissolution, these particles are in the form of primary particles. Because of the stabilizing impact of the SDAs, these particles are undergoing deglomeration. And from this study, we concluded that the faster than expected drug release can be attributed to the deglomeration of particles due to the stabilizing impact of release media. And lastly, we wanted to see what would be the impact of primary particles on the drug release of LAI suspension. So as I discussed that formulation FD and FE have a small particle size, and it is expected that these formulations should show very fast drug release. And from the drug release data, we can see that the drug release, although it is increased, but the discrimination is not much. So it is believed that the drug release from formulation FD and FE is slower than the expected drug release. So we conducted a similar study wherein the impact of release media was determined. And when we see the particle size data of sodium dodecyl sulfate, we can see that initially there is increase in particle size followed by a decrease in particle size. And when this media is replaced by PBS, there is significant increase in particle size on day 2 and day 4. And the possible reason for this could be the particle agglomeration that is taking place at day 2 and day 4. As we know that these particles are very small, there is very high Gibbs free energy of these particles, and these particles may be undergoing aggregation. So to confirm this, we did some microscopy experiment wherein we can see that initially the particles are present as primary particles, and on day 2 they undergo agglomeration followed by a decrease in particle size. And the slower than expected drug release could be due to the aggregation of the smaller particles. We also did some preliminary investigation in rabbits wherein the formulation FA was injected intramuscularly in the female rabbits, and this depot was removed at the 5th day of injection, and the particle size was determined using microscopy analysis. So from the microscopy data, we can see that the particle size was around 15 micron, which was not much increase for the formulation FA, suggesting that this formulation did not show any particle aggregation in the in vivo depot. Our future goal is to determine the particle agglomeration behavior in vivo for other formulations as well, and to correlate this data with the in vivo PK studies. To conclude my talk, this study highlights the critical role of particle attributes such as size and agglomeration in the performance of LAI suspension. Low energy sonication can be used to differentiate particle states into the suspensions, and suspension prepared with polymer from different vendor sources can have weak agglomerates of different strength, affecting the in vitro release rate. And particle-particle agglomerates can show faster than the expected drug release. And lastly, particles present as primary particles can show slower than the expected drug release. Thank you.